Okay, so let me start again. Okay, so a possible alternative title to the original one, why there's so few microbial species, would be why there's so many microbial species. And you could go either way. The first one just seemed a bit more fun to me. But really, it may be what determines the number of microbial species that I really want to talk about. And the system that I want to look at is not exactly this one, but something somewhat similar. So this is an effluent channel in Yellowstone National Park. There are actually a couple of hot springs up here, 2 different ones. And this is the effluent from the 2 different ones, of course, that mix together sort of, or come close to each other. And you can clearly see that, I mean, there just looks likely that there are different species here and there. And they're probably doing different things. So certainly, I don't actually know exactly what's going on here or what's going on at all, really, but the green is certainly cyanobacteria and or algae. Not sure what the red is. They do have iron oxidizing bacteria around, so it might have something to do with that. But in any case, they're doing different things for certain. But what I'm more interested in, what's going on if you just go downstream on, say, this guy, what sort of species you might see. So the green stuff is basically doing the same thing along here. It's just at different temperatures as you go downstream. Okay, so I thought I'd start by talking a little bit about species in microbes and how one might determine such things. So typically what people do is you sequence some gene, often 16S related. This is not that, this is actually a different gene from just Synechococcus related to photosynthetic machinery. And you get a bunch of different sequences, and then somehow you divide this up into different species. So the procedure that people will use is choose some well-conserved gene that you'll find in everything you want to look at. Say this one is typical, and then sequence this gene for lots of different organisms. And you decide, well, if the sequence of this gene for one organism is close to another one, then they're in the same species. 97% maybe is what you use. It's easy to do this in mass, but it's a bit crude. I mean, so I just got this off the net. Of course, it must be true then. But this is actually over the entire DNA sequence, that these are 96% similar rather than just a single gene. So 97% or any other numbers is arbitrary, but it's convenient. And what does it mean anyways? So here's some actual data. This is actually not where I want to go with this talk, but I thought it'd be interesting just to mention it anyways. So this is again, I think PSAA data. So they sequence a bunch of these genes from some population, and then they start binning them together depending on how close they are in sequence. And this is the number of bins that you need, or some minimum number of bins, say that you need to do this. And the idea that these guys, so I'm working with Dave Ward, who's interested in just deciding exactly what are the species in this particular system. And together, Fred Cohen has been involved in this, and other people too. And so they're looking for something called an ecotype. I don't want to get into that, but so my interpretation, and this is just strictly mine, one way to look at this is you're looking for some sort of clustering, and you get a gene where, well, you get a bunch of bins, and suddenly at some place, more or less everything bins by itself, or you get lots of little bins. And so maybe this is a good place to cut off if you're just doing it, trying to do this arithmetically somehow. There's still a lot of problems with this, but maybe this is the way to go. So anyways, our system is this one. So this is the effluent channel of a place called Mushroom Spring, and Dave Ward and co-workers have been looking at this for decades now, trying to decide exactly what are species, how do you decide what's a species and what's not, and particularly looking at cyanobacteria and how they change as you go down this effluent channel. So this is, of course, summer-ish versus winter. This section is up there. That's me supervising a graduate student. By the way, that's Michael Cool back there, if you happen to know him. So the question is, I mean, can you actually decide, are there actual species there? Is it just a mishmash of stuff? And how do you just, what determines where these species are? So this is old data, but it gets the point across. These are DGGEs. It doesn't, if you may or may not know what that is, but it doesn't really matter here. What's happening, so these are different temperatures. You take samples at different temperatures, which is basically different locations down this channel. And they're looking at PSAA, this particular gene again, and running it through this gel, which will separate things with slightly different sequences. And you see, you see different species at different, apparently, at different temperatures. So there's a discrete section where this species shows up, and then it's no longer there at higher or lower temperatures, et cetera. So it looks like there's not some sort of continuous variation of things, but there's actually actual real separations and actual sort of species. Now, and this is time of day, this is with RNA. Things are coming up and down at different times of day. This is up and down in the map, something similar. They have much better data now about this, but this gets the idea across. And then the next thing to do is to actually try to isolate as best you can these particular organisms, and they take them back to the lab and show that they're actually doing different things. In particular, they grow at different rates as a function of temperature. So they're actually, apparently, these guys are occupying different ecological niches. And this is a little bit different from where I want to go. So this is maybe the true title. I should say, basically, I mean, all these things are doing the same thing. They're cyanobacteria, and they're quite similar, apparently, in what they do. It's just that they operate at different temperatures, say, at different light levels. So they're all doing the same job, really. Okay, so maybe the real title should be something like this. Why are there different microbial species that do the same job in the same community, and how do you determine how many of them there are? So, I mean, there are different ways. You can have different species that do different things, and a lot of people have discussed that here. But this is not what I want to talk about. I want to look at, so in various situations, you seem to have a bunch of things doing the same thing, basically, and why is that the case, and how many of them are there? Why aren't there more or less? Okay, so I want to use as, for part of this talk anyways, as a proxy for a biofilm, a chemostat, which is actually not too bad of an analog. So here's a chemostat. What happens, you have a tank, well-mixed tank. That's different from a biofilm, but that's okay for our purposes. You have a well-mixed tank, you have stuff coming in at a certain rate, and stuff goes out at the same rate. So this is something like a biofilm. You have a fixed community, you have stuff coming across the top, some supply at a constant rate, and it also carries things away at a constant rate. Okay, so here are the equations. For the simplest possible equations for such a thing, you have a bunch of populations, and let's say you have one limiting resource. So the populations, they grow at some rate, and then they get washed out at some rate, and the substrate is used at some rate with a yield coefficient here. You have an input rate and also a washout rate as well. Okay, so what we're interested in is what happens over long times when you get to an equilibrium or something of that sort. The transients are gone, and then you get equations like this, and it's really the top equation that's interesting here. So if this thing is zero, you have two choices. Either the population is zero, it goes extinct, or its growth rate is fixed by the rate of the chemostat. Okay, so you don't get to choose your growth rate here. It's fixed by the environment. You have to grow at the same rate that the environment is telling you to grow. So the possible solutions, you can have either extinction, everything's extinct, or you can have generally one thing that is non-extinct. So for one of these J's, this equation can be satisfied. You choose S to satisfy it. But then for all the other equations, it's probably not satisfied, so X has to be zero. So you generally get exclusion. Actually, the one that wins, the stable solution, is the one that satisfies this term for the smallest value of S. So you drive the food level down as far as possible, and If you can drive it down below where everyone else needs it to be to survive, then you wipe them out and you win. Okay. So this is exclusion, and people wondered about this because there's more than one species out there. Why are there so many species? You have the paradox of the plankton, where you're out in the ocean and a lot of people are doing basically the same thing, but there are a lot of different species. How do you get around this? Well, of course, one answer, which doesn't really answer the paradox of the plankton, is you have a division of labor. So different things can do different things. You take your food and you turn it into CO2, but you do it in a number of steps, say. So you have different species for each of those steps. But what about the case where you just have a single job that you're interested in? So you have a bunch of species doing the same thing. Well, it's still easy to get around the issue if you have variation in time or in space. So if you have variation in time, you can have a species that does well in the environment at one time, the environment changes, and another species does well in a new environment, and it's easy enough to get them to coexist. And similarly, if you have variation in space, you can have something that does well here and something else that does well there. Okay, well, so here's the title, Why Are There So Few Species? If you're in a continually changing environment, why don't you have, say, a species that does best at each possible place going down the stream at each different temperature? That's not what we observed. We observed just a few of them. Same for time. Okay, so I want to divide this into two pieces, one temporal and one spatial. And let me start with temporal variation. So this is a graduate student, Shane Nowak, and he went down to this hot spring in winter and summer. This is Mushroom Spring in the background here. By the way, this is a famous spring. It's basically where Taq polymerase came from. This guy back here. It's been studied for a long time. People know a lot about what's in it. And let's go to the chemostat again, which is, I think, a useful simple model for looking at time variation. Won't be able to use it for space variation because it's well mixed, but it's still good here. And so what we're going to do is we're going to introduce a periodic environment into our chemostat, say temperature or light or something like that, something that varies in time. And what you get now is you get growth. Well, growth is modulated by how the environment changes in time. So there's a special function, f, that tells you how well you can deal with a given temperature. I'm just going to call it temperature, say, but it could be some other environmental effect. And so you just multiply your growth rate by this function, f. The choice of f amounts in this model to a choice of species. Okay. So just for future reference, note, we now have a couple of new time scales in the problem. One is from this environmental condition, the period of that, and one is, now this thing is no longer 0, it varies. It's sometimes positive, sometimes could be negative. So this is the growth rate, which gives you another time scale. It's the time, if you change the environment, how long does it take for this system to react to it, basically. Okay, so I mean, the idea is, well, if you have a changing environment, it's easy to break this exclusion and get more than one species to coexist. But that wasn't really what we were thinking about when we started this. Instead, we wanted to look for signatures of time variation in the species that were being recovered, or putative species that were being recovered in this map. What from the pattern of species could we say maybe came from time variation? So that was our thought process. So what we did in this model is we said, well, can we find an optimal species somehow? So F represents the species. You have to constrain it somehow, otherwise you just make it as large as you can. So we constrain it this way. F is non-negative, and there's sort of a total amount of effort you can put into any given temperature. So I mean, this is we have some. I mean, this is obviously not a reasonable biologically. You need more than that. So we have some danger of becoming, of just doing a mathematical problem becoming irrelevant, but I think in the end we do get some interesting information out of this anyways. So here's a few examples. You could have the red, green, or blue species that like different temperatures, say. So T is always, you fix it at the beginning, and then you just look for the optimal species F. So the issue is, well, it's true that you can break exclusion by introducing this time variation, and so you can get more than one species to coexist, but it may be the case that really, in the end, there is an optimal species that can exclude everything still. So you get back to exclusion. And what can it look like? Okay, so here's the question: is there an optimal species that, for a fixed capital T of T, outcompetes everything else? Can you find an F optimal so that no matter what other F you choose, as long as it satisfies those requirements, it gets excluded, so x1 goes to 0. So we don't know the answer. This is a mathematical question. Now, we don't know the answer to it, but it seems like it may be yes. It is possible to do that. So we're back to exclusion. So let me talk a little bit about the math. So we can simplify this in certain cases by decoupling the time scales, either looking at small period in capital T is a period P, so very fast fluctuations, that's like fluctuations in the cloud cover, or very large ones like seasonal fluctuations. So temperature varies a lot. Actually, light is the thing that varies quite a bit. Really, in these hot springs, the temperature doesn't vary as much underwater there. And then if you look at the intermediate case where the fluctuations and the growth response time scale are the same, you don't get a decoupling. So that's harder. I want to talk mostly about this case. This is both interesting, the results, and more tractable, and I'll mention what happens in the other cases a little bit too. So when you do this, basically, so you have F of T is changing very slowly now compared to the dynamic time scale. So you can kind of assume, if you do the asymptotics, basically what happens, you can kind of assume that this is fixed almost, and then solve at each given time for a fixed F. So that makes it easy, relatively speaking. So in this case, we still can't do the problem, but we can do the following. We can ask the following question: is there a species that can invade a pure culture of any other species? So you take any possible species, you grow it up into nice pure culture, you introduce a little bit of the optimal species. If there is one, can you find one which will always successfully invade? And the answer to that is yes, you can do that. There is such a species, and it is unique. There's only one of them. It turns out to be a solution of a particular variational problem. Just coincidentally, I just mentioned this. So you want to minimize. So in a steady case, you minimize S. In this varying case, you actually minimize the log of S. This is essentially the geometric average, not the algebraic average, which is kind of interesting, but neither here nor there really. The answer is important. The optimal species, basically, it weighs a given environmental condition, how much effort it puts into that by W. W is the density of the environment, so which is basically how often any given capital T occurs. So if capital T occurs a lot, you put a lot of weight there, and if it occurs little, you put less weight there, and you just do it exactly in proportion to how often a given environmental condition occurs. Notice this is a weak dependence on this thing. It only depends on this density, not on the details of how these fluctuations actually go. That has to be the case because the timescales are independent of each other. The growth can't really see, the species can't really see the variations in environments, so they have to decouple this way. There's a little numerical example. I've got two species here. We have a sinusoidally varying environment, so it varies between 57 degrees and 63 degrees or something like that. And here are two different species, one minimizing log of S over on the average, one minimizing S on average, so geometric versus arithmetic. Very similar. You put most of your effort at the ends where the environment spends most of its time. But the geometric one wins, and this blue curve would go to 0 after a while. So it not only is able to invade, but it's actually, numerically, it seems to be able to wipe out anything else. It seems to be a true optimal species here. Okay, so I mean, I think we have an optimal species, one that will wipe out any other species in this case, even though it's possible to get things to coexist. There is one that could come along and get rid of everything else. It is accessible in the following sense, or seems to be. You can find, you start from any given species, you can find a string of other species that are close to each other that start from the original species and end up at the optimal species, and each successor can invade the predecessor. You can show that. And numerically, it seems to be the case that each successor actually wipes out the predecessor as well. So, I mean, you might think then that we've lost exclusion here, and that's what this would seem to indicate. Let me skip that one. Okay. So just some general observations about the model. So here's the ones I kind of went through for a long period compared to the reaction time, the growth time. There seems to be an optimal species, and only the density of the environment matters. Just to let you know, in the case of a short period, you also can answer this. There is an optimal species. Again, it's a delta function. So what happens in the short period is the environment fluctuates so fast that your species isn't able to react to it. It can't respond to it. It's too fast. It doesn't really see the fluctuations. So what it should do is just sit on the most common environmental condition. So put all of its effort there. Of course, that's not realistic, but what this is saying is that rapid fluctuations don't determine the niche width here. Again, notice only the density matters, not the details of how you do this. And it's the same reason, mathematically speaking, the periods are so far apart that they can't see each other. The intermediate case is also interesting. So there we only have numerics. In this case, the response time and the fluctuation time do understand each other. They can see each other. So not only W, but the actual details of how you vary time, how you vary environment actually can matter. And what the model predicts something like a clock. So so you have a regularly varying environment like day-night cycle. That's roughly on the time scale of growth of a cyanobacteria, say. Cyanobacteria knows it's coming, and it can respond to that. So it can get ready for good times and for bad times, say. Okay. So we wanted to test some of our predictions against the actual system. So this graduate student, Shane, did the following things. First of all, he went out and measured light intensity year-round. I'll show you the plot of the data in just a second. And he threw that data into the model. So the model says, given capital T, we can predict an optimal species. So he did that. He measured T of T and predicted an optimal species. And the second thing he did was he went and looked at some of these organisms that are growing up at the top of the mat where the light is similar to what he measured, and he grew them up in the lab at various light intensities and measured their growth rates. Then you can predict. So this is the model prediction of what the optimal species would be. And this is the prediction. This is what is actually there, and you can compare. Okay, so here's the actual light data. This is 1 year. You can see that there's obviously a long period and an intermediate period are these spikes. That's each day. So there's a mixture here of long and intermediate, and plus short, but the short doesn't really do anything. This is the light meter. It's actually embedded in this stone here. and this is our effluent channel. We had to put it in a stone because we were supposed to disguise it so people couldn't see it. And there's a cord that runs along here to a logger, which is weird because people aren't allowed back here anyways, but we still had to do it. Okay, and here's the process I don't really need to go through. This is actually a cross section of this mat. So you try to grow up individual colonies, and then you sequence them to make sure you have the right species. And this is the growth apparatus. So these black sleeves change the light intensities, and he's got a water bath to keep the temperature right, and he's bubbling it through CO2 and stuff like this. And these are various different runs of this thing. And here's the sort of growth curve you get: growth versus light intensity. Interesting, just an interesting thing actually, as long as we're here. So if you notice, you look at the changes in light intensity from winter to summer, it's actually about a factor of 4 difference. There's a huge difference there you might not realize. And this particular intensity, this is a very large intensity. I'm not sure what the light intensity down in this room would be, but it's probably somewhere down here. So the light during the day in the summer is very large, very high. And he wanted to get up to that height. So this is only going to 500, but I'll show you 500 microeinsteins. But I'll show you more data in a second. So he had to build a special apparatus to do this with very high intensity lights, and we weren't sure how to do that actually. But this was happening a few years back, which shortly after there was this ballot initiative in Montana where this was going on, which legalized marijuana. So we were hoping, well, because of that, there would be all sorts of fancy lighting gear that we could get hold of. Turned out even for those guys, the light intensity was that we needed was too high, so we had to build a special mechanism anyways. Okay, anyways, so this is the comparison of the model and the data. This is the model output over here. So we took that light intensity data and plugged it into the model, and we also needed a growth frequency, so we use a few different growth frequencies. Roughly 1 to 3 days is the right one here. So you can see the curves for this guy. So this is the predicted F. How much effort roughly you'd put into different light intensities for the optimal species. This is the density function just for reference of how often you got a particular light intensity over the course of a year, relatively speaking. And then this is the curve I just showed you. This is actual growth of one of these species that lives at the top of the mat. So what we're comparing is this curve versus this one, which we were actually fairly pleased with this comparison. The important thing is that both of these predict a cutoff in this range. So even though the light intensity gets quite higher in summer, it gets all the way up here, the species that we're seeing at the top of the mat cut off well below that. And the reason, so it's around here basically. The reason is, roughly speaking, so in this range, this is high light roughly in winter. So up to about here are light frequencies you see all year round. These high ones you only see part of the year, and these only in the summer. So what the theory predicts is that you shouldn't really want to waste time dealing with these very high light intensities, and you should cut off down here, and that's what we got. Of course, there is an issue here. This one goes off to infinity or whatever is their predicted one, and the real one goes to 0. Well, of course, it has to go to 0 because as the light intensity goes to 0, you run out of energy. That's not in the model. The model only deals with, it doesn't deal with the energetics at all. It only deals with the frequency that you see. So because of that, the model doesn't know that it can't grow up here, so it puts a lot of effort there. So we're not so worried about this. What we like is this part down here. By the way, these particular cyanobacteria do have clocks. as well. I'll show you that in a second. So just a little word about these intermediate oscillations and what I mean by clock. So here are two possible choices of capital T of T, the dotted one and the solid one. They're chosen so they both have the same W of T. They both spend the same percentage of time at each particular temperature or light condition. They just do it in different order. And if you plug this in the intermediate case, you get different answers for the optimal Fs. In particular, for this jagged one, it's very clear that this guy has a clock. It's getting ready for good times or for bad times by basically setting its effort to 0 right around here. 59 and 61 are kind of the interesting temperatures here. That's what we mean by clock. Here's evidence of the actual clock in the cyanobacteria. You have certain sets of genes that get turned on during the day. This is light here, and others that get turned on at night. So it's definitely there. Okay, so that's time. Let me talk about a little bit about space as well, briefly. So this is work with Jack Dockery and Hal Smith. And so here again, what we're interested now in, as I should show the data again probably, is how does species change as you go downstream? Excuse me. Okay, so we set up a model of our biofilm now. We can't use a chemostat anymore because we don't have a well-mixed system. We're interested in what happens as things change in space. So we set up our model to have one particular key element of a biofilm, and that is competition for space. So, for example, this is a cross-section of a lab-grown biofilm. There's an active layer on top where I think oxygen is being used, and below it's depleted. If you want to use oxygen, you have to get into this layer somehow. You have to be able to compete for that space. And if you can't, you'll get pushed out one way or the other, and you'll be in trouble. This is the cross-section of the mat again, and we're interested in this very top layer where there's light. Below that, the light is depleted. So if you're interested in light, you have to manage to grab some space in there. You can have lots of layers, but in each case, and nevertheless, if you like to use this as your electron acceptor, you have to make your way into a particular space and be able to compete for space there. The second thing that we added is some mobility. So if you have an issue, if you have a distribution in space, of course, there's always the option to move around and try to find somewhere else. There is actually some evidence for mobility within biofilms. People don't really look for it that much, but it's probably there. In this particular case, this is looking at up-regulation and down-regulation of various things in biofilm, and let's see. The star is the biofilm, the other are planktonic cultures, and the one thing that's clearly up-regulated here has to do with Tritium motility. There's other evidence as well, but it's just one example. Okay, so here's our model. We have a one-dimensional space labeled by X, say that's distance along an effluent channel. We have an environmental condition, say temperature along the channel. So we want to vary it linearly, so the gradient is constant. There's no special preferred area in one place over somewhere else. And then we have a continuum of species. Capital X is volume fraction at time t at location x, and tau is the species label. So for example, tau might be the best temperature for that particular species to live. And its growth is modulated again by a function of this form. Tau is your best species, and then you subtract off the actual best environmental condition, and then you subtract off the actual environmental condition. So F is largest maybe at argument 0, and it gets smaller as the argument goes positive or negative. So we have this guy, and they all diffuse a little bit. The question is, so we have a continuous species. The question is that what you actually see in the solutions, or does it discretize in some sense into just a few finite species by itself. Okay, so here's a possibility for how these environmental response functions look like. So this is response. This is your preferred environmental condition. This is the actual condition at a given location. And so you could get something like this, a Gaussian, or it doesn't really matter what you use. For the most part in this model, we also used non-symmetric things. There's an interesting story about this particular function, but it doesn't really matter here. It just, I just want to make the point, pretty much anything, you get the same, seem to get the same answer. And here's the equation. So you just take those elements that I described a couple slides ago, and you put them together, and here's what you get. So this is the diffusive piece. This is the growth piece, some growth rate modulated by this response. And then this is the piece that gives you the competition for space. And so you determine this by this condition, this constraint. If you set at a given time at a given location x, and you add up all the species that are there, so you do this integral over the species, it has to be equal to 1. So if you integrate this equation over tau, you get an expression for c. It turns out to be this thing. So okay, so this is kind of a scary looking equation. It's an integral differential equation, but it's actually not so bad as it looks, because this integral, the equation is really in t and x. The integral is in tau. So this integral doesn't interact directly with the rest of it, so it makes it a lot easier to deal with. Okay, so before I get to the general case, let me say a little bit about a special case. Suppose instead of a continuum of species, we just take 2 species. Can they coexist? Okay. So we have these are the equations now in the case of just 2 species. Rather than that integral differential equation, we get 2 PDEs, 1 for each. It looks similar to the one before. And you can actually combine them because x1 plus x2 add up to 1. They're volume fractions. And when you do that, you get a single equation for one of the species. This is a Fisher equation, kind of well known in mathematical biology. It's a little bit different because this coefficient, well, it's not constant, first of all, but more importantly, it changes sign. So if you know the Fisher equations, they support traveling waves often. But if with this coefficient here changing sign, basically you can sort of get traveling waves that want to crash into each other, and so you get equilibria formed. And this thing has actually been studied in biological contexts. And what you get is generally speaking, if D is not 0, you get a stable equilibrium where x is between 0 and 1, meaning both species, the 2 species coexist. So what we wanted to know, we want to know what, so this is 2 species, we want to know what happens when there's a continuum of them, lots and lots and lots of them. So what we actually want to do is take these 2 species and crash them together, let their preferred environmental condition become similar, and see what happens to this coexistence. There's 2 possibilities. So you have coexistence. You could have either they sort of separate space, so one species occupies the right-hand side, one species occupies the left-hand side, and that suggests that you're going to have that your continuous species will break down into a finite number, or you could have the 2 species basically occupy all of space together, and it's actually the second one that happens. The asymptotics are doable, and what you get is basically each species volume fraction goes to 1/2 through your domain. So that suggests that these things will coexist. In fact, that is what happens. Numerically speaking, we can't do this problem analytically, even the instability problem. So we have numerics, and here they are. Okay, so let me explain what this is. So we solve the equations, and here's at a given, let's see. So this is we look at a particular species and see how it distributes itself in space. This is what it looks like. and we look at a particular location in space and see what the distribution of species are, this is what it looks like. These are actually identical for this choice of F because X and tau are symmetric. But even if you use something else, the picture doesn't change too much. The interesting one is this one. So let me explain it. So this is space here, regular space here. This is species space here. And so as you travel in, if you look at a location in space, you get a range of species, these guys here at this location. And as you move along here, you get a same, you still get a distribution of species. It's just been shifted down, and this shifts continuously. So basically, every species, possible species, is out there, and it's located in some region close to its optimum. You get the continuum of species here. So this is numerical. It seems to be stable. In other words, there's nothing inherent in this model which will cause these things to break up into a finite number of species. That's our conclusion number one. So now, well, let's start pushing this thing. We'll jab at it and see if we can do something to it which will cause it to break up. So here's one possibility. We start playing with our environment. So so we make some conditions better than others, inherently better than others. So for example, you could do something like this. So certain environments are just better for growth than others. And this is with a long frequency interval here in space. And this doesn't do anything. You still get basically the same thing. But what it turns out will do something is our short length instabilities. And one in particular important one is a boundary. So a boundary introduces a short length scale. And here, so before we had basically an infinite domain, now we have a finite one. And we have boundary conditions on the ends. In this case, no flux boundaries. And what that does, that's a short length scale perturbation, which makes the guys on the end better off because these guys on the end, they're competing against these ones to the right, but not to the left. So they have an advantage that way, and they can start to take over some space. And same down here. So you can see this particular species starts doing well in this region, and it gets better and better. And as that happens, so it starts wiping out nearby species. And so effectively a new boundary gets set up here because this thing has wiped out all these species. And this particular species can do well starting from about here. And it starts taking over its local space. And the whole thing spreads inward. So you get this, you get this thing to drop off into a bunch of, into a finite number of species. Okay, so just to summarize, so what we see for evenly spaced for evenly varying environments, we don't see any kind of inherent tendency to discretize. But if you have a perturbation of a particular length scale, then you do get this. And the length scale is this. I didn't say this in the previous slide, but the length between these various species is this number square root of d over r, which that's the length that something can diffuse before the reaction time, response time becomes important. So if you have variations in your environment on that length scale, and you always will, then you'll get discretization into species, and the length between species is basically on that scale as well. This has been seen in other places here, but as well, but it's very clear here. So there's a particular length scale. D is diffusion. R is a growth rate. That gives you an environmental length over which you expect to find a discrete species. Now we can't really test this because we don't know what the mobility is in our particular system, but it's at least consistent. So it's reasonable compared to what we see in the Yellowstone maps. Okay, so let me finish up. These are the various people that I worked with. I want to thank staff and organizers here, especially, and I'll take any questions.
in your third set of results that you talked about, which was this spatial separation or not as the case may be, there has been some work that's been done where there's an assumption that species interact with each other in space, but in a non-local way. So the argument is that they interact either because they essentially they live somewhere, but they can move around, and so they sample a local space, or it can be that they interact through some kind of resource which is depleted locally, and so therefore they're aware of species which are nearby in their open space. And what they found was that if under certain circumstances it breaks up into individual species, and sometimes not. And it depends very critically on the shape of the interaction cone. So if interaction cone is a Gaussian, then that's kind of a critical case where you need to add some further kind of noise in order to, or heterogeneity in order for it to form patterns. But if you have anything that, any interaction kernel that has negative Fourier components as kind of the general result, then you end up with separation into separate. Is that?
Yeah, so I think I'm aware of what you're talking about, and we weren't very happy with that because it depends on very delicate properties of the tails of F. So that's part of the reason why we went to this model, which instead of interaction, we have the actual mechanism of competition here, which is spatial competition, and we no longer have that issue with the tails. The form of the tails doesn't matter. There's none of that kind of sensitivity here.
So what I'm thinking of, it's not actually the tails, it's just whether or not there's a negative Fourier component. You can get that for any kind of kernel which is sufficiently abrupt.
Oh yeah, so, well that's what I mean by tails. You need sufficiently abrupt to fall off.
But sufficiently abrupt is not, I mean there's many differences. It's quite robust to,
well, yeah, I mean if you don't want to call it tails, that's fine, but it is delicate to how things fall off. So you take a Gaussian versus something that falls off a little bit faster than a Gaussian, and you get a very different result.
What you showed in some of your pictures is that many of the species had only very narrow temperature range, right, between 58 and 61 degrees centigrade, for instance. But I can imagine that many of the species in this mix have a much wider temperature range, precisely because there's a lot of fluctuation in temperature in space and time. Did you investigate this, like how your model assumptions on the width of the temperature response?
So you're asking these are the temperatures where they were found, or you're asking what temperatures they can actually grow in?
Yes, exactly.
No, so I would assume that's quite a bit larger, but I don't know that anyone's actually done growth measurements for different temperatures. We've just done them for different lights, and I don't think it's been done for different temperatures. So those fluctuations of that range are roughly what you see over the course of the year. About that size.
Okay, so it is rather narrow temperature fluctuation in the field that you measure.
Yeah.
Are only a few degrees.
Well, it's a bit more than that, but not a lot more than that.
And would your model results be strongly altered if you would have the same bell-shaped functions, but say much wider?
Are you talking about the spatial ones?
No, the temporal line.
Yes. So yeah, the temporal ones. I mean, we don't have a particular. So the temporal ones were applied to light. We didn't try to apply them to temperature because I mean the temperature range is fairly flat. I mean the W is fairly flat there, and it would be interesting to try to do that. I can't remember now exactly why we didn't. But I mean, basically what the prediction that you get, I guess, is the range, the niche range should be roughly the same as the fluctuation, and we're not too far off there. The fluctuation range is a bit larger than what we see, but not a lot, a couple degrees more. Yeah. I'm trying to remember. I mean, I don't think we tried to predict exactly which how the species should arrange themselves in this case. but the width prediction is about right.
Your temperature response stuff, the first part of your talk, the results that you had, those are assuming that the temperature response function had integrated to 1 or integrated to a constant value. Does it change your results much if you make different kinds of assumptions on the constraints of the function?
I'm sure it could. You get fairly unrealistic things like, shapes like this. So you could and we thought about this, putting a penalty, say, so you don't have two different. There should be a penalty maybe for having these two different lobes or something like that. So, we only for our particular model we get one optimal species, but maybe if you start adding more constraints, you start getting a few extra species or something like that. It's very possible. Yeah.
One of the results was that the under certain circumstances, the optimum response is just a single Dirac peak, and it's kind of difficult to imagine how that could arrive biochemically. So there has to be some kind of constraint on how bendy the response can be.
Certainly, yeah. So, but our interpretation of that in this context was that that was in the case where you have very fast fluctuations. So our interpretation was, well, the fast fluctuations aren't going to determine what exactly the F should be. And I think that's basically the case anyways. Your organisms just can't respond to very fast fluctuations anyway.